Hi, this is Sweta, and our team members are Meghna and Vishnu Vandana. Our project title is Agent Based Epidemic Simulation Model. In this project, we have done some analysis and results. Coming to introduction, this project main aim is to investigate the spread of infectious disease within a population, and we use various parameters in this project. They are like number of agents, height and width of the grid, probability of transmission, death rate, recovery days, standard deviation of recovery days. And we use visualization tools in this project like Mesa framework, IPy widgets, bokeh, and panel libraries. Coming to main model description, the environment is represented by two-dimensional grid on which the agents are put. There are three possible states for agents, like there are susceptible, infected, and remote. Where susceptible is represented by blue color, infected is represented by red color, and remote is represented by green color. And also a normal distribution with mean age is 20 and standard deviation of 40 is used to generate the beginning age of agents. Each agent has ability to travel randomly over grid and communicate with agents nearby. And contact with an infected agent and susceptible agent may result in transmission of disease depending on their health and prevalence value. Coming to disease dynamics, when an agent contracts the infection, they are infected for a predetermined amount of recovery days. Depending on the death rate parameter, they either recover or move into remote condition after this time. The eliminated agents are no longer involved in spread of disease. Then coming to main code, we have installed libraries like Mesa, IPy widgets, bokeh, and panel libraries. And coming to main code, we have imported agent model, random activation, multi-grid, and so on. And we have considered constants like number of agents we took as 500 and width we took 50 and height is 50. And in this project, there are three classes. This is status, this is agent, and this is model. And also in disease agent, we have variables like self, unique ID, model, initial status. And in this disease agent, we use functions that step function, move method, and try to infect, recover, or die. These are four methods in disease agent. In disease model, we also use various parameters. And coming to disease model, it defines the model and parameters that control its behavior. And in this disease model, it total controls the entire population behavior, and it initializes the population of agents on grid and sets the initial state of each agent to susceptible, except for one agent that is initially infected. At each step, the step method is called on each agent in a random order. The data collector object collects data on the number of agents in each disease state at each time step. Coming to count functions, there are like count susceptible, count infected, count recovered, count dead. These functions are used by data collector objects to collect data on the number of agents in each disease state. These functions use list comprehension to count the number of agents in each state and returns the result. Overall, this code defines an agent-based model to disease spread, and this MESA library is used to simulate its behavior and collect data on its dynamics. And next part is will be continued by my friend Meghna.
In this project, we are using two functions for plotting the graphs and the plots. Those are plot states bouquet and plot cells bouquet. In plot states bouquet, we are using model and title as the parameters. This function is used to create a line chart that will display a number of infected and the recovered agents that are displayed by red and green respectively. And this function retrieves the data from the data collector object, and it stores the count of agents in each disease status at every step. And in plot cells bouquet, we are using only model as one of the parameter. This function creates a scatter plot that will display the location of all the agents and the health status. Like healthy will be displayed as blue, infected as red, and removed as green. And coming to this code, we are using IPY widgets for the sliders. And the sliders that we are using over here are the parameters that we are considering for this project. The parameters that we are using are number of agents, width, height. P trans is the probability of transmission rate from one person, from one agent to other agent, and the death rate, recovery days, and the standard deviation of the recovery days. These can be adjusted by the user, and these are user friendly. Like this interface allows the user to adjust these according to the demand of their users. And after running this code, we'll get these sliders so that we can just adjust them accordingly. So the weight will be adjusted, the probability of transmission rate is adjusted, then death rate could be around 0.24, and the recovery days are around 39, and the standard deviation will be 23. And after running this, and now we are importing over here different modules like agent and model. Agent for only a particular individual, and the model class is for all the individuals. Here we are using a random activation. We are importing it because the agents are moving from one cell to other cell by randomly. And the multigrade, we are using a 2D grade for our project. And data collector, we are using it for collecting and storing the data. And we are also using pandas, numpy for batch runs. Numpy is used for mathematical operations, and the time is used for measuring the time that is taken for executing the project. And we are also importing certain modules from bouquet and also panel, that are the libraries that are used for visualization tools. And now coming to the presentation that we have created, these are the initial parameters we have set to, like the number of agents are 500, the width is 20, the height is 20, probability of transmission rate is 0.25, and the depth is 0.01, and recovery date and the standard deviation of recovery date. These are the initial parameters, but which can be adjusted later on. And also the coming to the results of those sliders, probability of transmission rate, if higher the transmission rate, higher is the spread of the disease from one person to other person, and the lower transmission rate will obviously slows down the transmission. And coming to the death rate, higher the death rate, decline in the number of infected agents, because once the individual or agent is dead, we are removing from our simulation. And lower death rate will obviously prolong the epidemic as they continue in the simulation. And the recovery days, shorter the recovery period, the faster is the decline in the infection rates, because they are recovering in less number of days. And longer recovery period will obviously result in the prolonged epidemic. Now the simulation graphs and all will be continued by my friend Vandana.
Hi, this is Vandana, and I'm going to explain the graphical representation of the code. So here is the graphical representation of the code initially, and as it goes, this is the intermediate representation, and this is the final representation of the code graphically. So let's get back to the code again. So this part of the code, it represents that the plot states bouquet function is a utility function that creates a bouquet line chart to visualize the state of a disease model over time. It uses data collected by the model's data collector object and creates a column data source object that can be used to populate the chart. The chart has three lines, one for the number of susceptible agents, one for the number of infected agents, and one for the number of removed agents, which can be known as recovered or dead. The x-axis here in this graph, the x-axis shows the time steps of the simulation and the y-axis shows the number of agents. The function returns the bouquet figure object that can be displayed in a Jupyter notebook or can be saved as a file here. And coming to the next part of the code here, it creates a visualization of the simulation using the bouquet library. The code initializes a disease model object with certain parameters based on user input and then runs the model for a specified number of steps. Let's run the code here. Pause చేసా.
ఏమైంది?
Screen recording కూడా sorry, there was a small issue with the code. And why
And I'm now running the last part of the code here. And as you can see, the graph is changing. So here, for each step, it generates two plots using bouquet. One plot showing the number of agents in each state that is susceptible, infected, recovered over time, and another plot showing the grid of agents with different colors based on their state. The two plots shown in here, this one and the other one here, are then displayed side by side using panel, and the process is repeated for each step. As you can see, the clear output function is used to clear the output area after each step, and time.sleep is used to pause the execution for 2 seconds to create an animation effect, as you can see here. So the graphical representation happens, and this is the final output. And as you can see here, as you change the parameters here, the graph also changes. It's not the constant one. So by simulating various scenarios, the model offers valuable insights into dynamics of disease transmission and the effectiveness of different control measures. And the future work can be incorporating spatial and temporal heterogeneity, model difference disease characteristics, investigating the impact of public health interventions, and integration with real world data. Here are some of the acknowledgements and references. Thank you.